Welcome to Provitalks, where nature and science blend to inspire you. Discover the latest in technology, sustainability, and trends in the cosmetics world, thanks to Provital. Let's care together for our industry's commitment to the people and the planet. Provital. Do care.
Hello everyone, and welcome to this edition of Provitalks. In these next two episodes, we'll go deep on the science and technologies behind cosmetic research and development. Specifically, we're going to talk about the newest advances in cell senescence and how can we apply artificial intelligence to bring new cosmetic claims to the table. This is Clara Vigo speaking, and I'll be extracting all the information I can from the brilliant mind behind the development of Altheostem, our latest launch, and a cosmetic ingredient that perfectly exemplifies these two technical concepts. Let me introduce you Dr. David Manzano, Provital's R&I Innovation Manager, and our expert today here in Provitalks. Besides his PhD in biochemistry and molecular biology, David has worked on different research topics, including the regulation of plant metabolism to produce natural compounds with high added value. He has also published several papers and book chapters in relevant peer-reviewed journals, and he's presented his work in different national and international conferences. Now, this broad experience he's got in the private sector allows him to develop cutting-edge research projects for the beauty sector at Provital. Well, welcome, David.
Thank you, Clara, for this wonderful introduction and for the invitation to this edition of Provitalks.
My pleasure. I'm sure your knowledge on research and innovation will be of great use to our listeners and for the topic of this episode. So thank you for taking the time to be here. And let's get straight to the topic, right?
Sure, let's go.
I mentioned at the beginning that we would be covering the newest advances in cell senescence today. So David, what do you think if we start by, briefly introducing the concept of cell senescence?
Of course. As many of you probably know, cellular senescence is one of the hallmarks of aging, together with other well-known processes such as genomic instability, altered cellular communication, or stem cell exhaustion, to name a few. But we can describe senescence as a cellular state that is triggered as a response to damage inputs, such as oxidative and genotoxic stresses, telomere shortening, DNA injury, or mitochondrial dysfunction, among others. In addition, cells can enter a senescence when they reach their replication limit capacity, also known as the Hayflick limit. In any case, whatever the damage input is, cellular senescence results in a multiple phenotype consisting of morphological changes, irreversible growth arrest, resistance to apoptosis, and the release of a complex cocktail of inflammatory factors known as the senescence-associated secretory phenotype, or SASP. These factors can degrade the local tissue environment and induce inflammation in tissues and organs. Now, the normal role of the SASP is to restore tissue function by stimulating neighboring cells to engage in tissue repair and by attracting immune cells to eliminate senescent cells. However, when the damage inputs exceeds the repair capacity, what happens during aging, senescent cells accumulate, worsening tissue malfunction and also transforming the healthy cells into the senescent cells as well. That is why senescent cells are also named as zombie cells.
Yeah.
I would like to highlight here that in human skin, senescent cells have been correlated with visible signs of advanced age, such as wrinkles, lack of elasticity, and perceived age.
Okay, but correlated how exactly? I mean, how can then we revert it?
Okay, so for the first question, correlated how? Let me explain that senescent cells are more prevalent in aged human skin compared to young individuals. And there is evidence linking senescent cells to other biomarkers of aging. In a very interesting study conducted with skin biopsies from 178 participants, aged between 45 and 81 years, the counts of senescent cells were compared to local elastic fiber morphology. facial wrinkles, and perceived facial age, while accounting for chronological age. The results showed a significant association between the numbers of epidermal and dermal senescent cells and age-related elastic fiber morphology characteristics. Senescent cells were also significantly associated with more facial wrinkles and higher perceived age. Participants with the lowest number of senescent cells appeared 3 years younger than those with the highest numbers, regardless of their chronological or real age. In conclusion, senescent cell numbers in human skin are indicative of both local elastic fiber morphology and the degree of aging visible in the face. The good news, however, is that cutting-edge research demonstrates that the genetic clearance of senescent cells extends the lifespan of mice and delays the onset of various age-related diseases. Additionally, pharmacological suppression of senescence has been shown to increase healthy lifespan and reduce the age-related diseases in mice and some species of monkeys, as well as it's able to restore homeostasis in senescent human cells. More recently, CAR T cells have exhibited senolytic activity, effectively reversing senescence-associated pathologies. All these findings underscore the significant therapeutic potential of strategies aimed at mitigating the negative effect of senescent cells. Finally, these strategies can be classified as either xenomorphic, when the objective is to suppress the SASP phenotype, or senolytic, when the target is the selective elimination of senescent cells.
So, to put an example that I know, Altheostem is a senolytic. Therefore, it will revert this situation by means of rebalancing the senescent cell population to an earlier age, then selectively promoting apoptosis of senescent fibroblasts in this case, and then restoring the extracellular matrix, right?
Exactly. What we have achieved with Altheostem is the selective elimination of senescent cells. So therefore, its effect can be classified as senolytic. We have proven its effect by different in vitro experiments, including senescent cell viability assays, quantification of the number of senescent cells, and apoptotic markers, as well as evaluation of the consequences of the active on the expression of genes important for the extracellular matrix formation.
So thanks to this senolytic effect of Altheostem, we can selectively eliminate those senescent cells and have a rejuvenated skin. However, as far as I know, Altheostem's way of proving and quantifying that selective mechanism of action, the senolysis, is what got us the recognition in the IFCC Congress in 2020. So please, David, could you tell us a bit more about the in vitro senolytic quantification for this active?
Yes, of course. We did so by different approaches. The first one was to evaluate the senolytic activity of Altheostem, assigning its effect on the viability of a model of senescence, which is the extrinsic senescence model in human dermal fibroblasts. As a control, we used the same lines that were not induced for extrinsic senescence, but were grown in parallel and incubated with the same concentrations of Altheostem. The results show that the viability of senescent cells was reduced when compared to normal control cells in a ratio ranging from 14 to 45% of reduction.
Impressive, almost half.
Yeah, almost half, exactly. These differences are also statistically significant for Altheostem, concentrations of 0.5% and above, and moreover, these results are correlated with the concentration of Altheostem in a dose-response manner.
Okay.
Now, that was the first test. The second one, to further confirm the senolytic activity of Altheostem, we addressed this time the quantification of the number of senescence-induced human dermal fibroblasts expressing beta-galactosidase. Now, beta-galactosidase is a specific biomarker of senescent cells. To this end, the senescent fibroblasts were incubated again without or with increasing concentration of Altheostem. And what we saw is that when the senescent cells are incubated with the Altheostem, there is a strong reduction in the percentage of beta-galactosidase positive cells. We reduced the number of the proportion of senescent cells by almost 43% at the highest assay dose. And again, these results are correlated with the concentration of Altheostem in a dose response manner. Now, finally, with the aim of understanding a bit more the underlying mechanism of the Altheostem senolytic activity, we quantified the capacity of Altheostem to induce the apoptosis of senescent cells. The effect of an apoptosis was analyzed by quantifying two different apoptotic markers and comparing the results to the obtained with control non-senescent cells that were grown in parallel and incubated with the same concentrations of Altheostem. The first of these markers was the exposure of phosphatidylserine on the surface of the cell, that is a biomarker of apoptotic early events. And what we see again is a very strong reduction and a dose-dependent induction of the apoptotic marker only in the senescent cells when they were treated with Altheostem.
Wow, so that was an extensive quantification for sure. But how is that senolytic effect contributing to the remodeling of those skin functions and components that you mentioned before? I mean, the ones that are affected by skin senescence.
Well, in this regard, we wanted to evaluate the biological relevance of this senolytic activity of Altheostem, especially in the context of its potential anti-aging application. For this purpose, we perform a gene expression analysis of relevant genes involved in extracellular matrix remodeling in human dermal fibroblasts. What we did is that we compared the gene expression levels of an senescence fibroblast treated or not with Altheostem. The results show a clear induction of genes involved in the formation of the extracellular matrix, including collagen 1A2 and ADAMTS2. Both genes are involved in collagen formation, hyaluronic synthase 3 and fibrillin 2 that are involved in the hyaluronic acid and fibrillin biosynthesis, respectively. And in contrast, genes involved in the extracellular matrix degradation, such as metalloproteases and the pro-inflammatory factor GM-CFS, are strongly repressed. So altogether, these results reveal the anti-aging in vitro effect of Altheostem.
Yeah, exactly. So all in all, Altheostem reverts that phenotype that was displayed by senescent cells. So it is promoting a younger and healthier skin. And well, David, I also know for a fact that such cutaneous effect was clinically proven both with an instrumental and
Yes.
an artificial intelligence analysis. But what do you think? We'll leave the technicalities of this part for our next episode.
Yes. We obtained very good results in our clinical study by analyzing instrumental parameters and complemented the results by evaluating the visual apparent age of volunteers using artificial intelligence innovative tools. But sure, let's discuss this in our next episode.
Yeah, that's better. Let's stop here for now. I think there is already enough technical information for our listeners. So thank you very much for this amazing explanation on skin senescence and senolytic advances in cosmetics research, David.
Well, it was a pleasure to explain our results, so thank you for the invitation.
Thank you. And to our listeners, we hope you've enjoyed this podcast as much as we did. We really hope you can find in our conversation a form of inspiration for your potential projects. And well, thank you all very much and see you in part 2.
Thank you for listening to this edition of Provitalks. We hope you got some source of inspiration to create and spread your own kind of beauty. For further information, subscribe to our podcast or visit our website at weareprovital.com. Provital. Do care.